if a cell is actually to exist and function effectively and efficiently, it must be able to control and regulate the way that the cell expresses the genetic information that is found inside DNA molecules. Now, what that basically means is the cell must be able to control the process of transcription. It must be able to turn on transcription when we need to synthesize proteins and turn off transcriptions when we do not need to use those proteins. And this process is known as gene expression and gene regulation. Now, prokaryotic cells, such as bacterial cells, can regulate gene expression by using special types of molecules, special types of proteins known as DNA-binding proteins. And DNA-binding proteins used by prokaryotes come in two categories. We have repressors and we have activators. So during the process of transcription, when we transcribe the DNA into RNA, the activators can basically activate transcription while the repressors can block the process of transcription and ultimately block the synthesis of proteins that correspond to those genes. Now, our activators and repressor protein molecules can themselves be controlled allosterically by other molecules, as we'll see in just a moment. So when we're discussing prokaryotic gene regulation and expression, basically the model that describes the regulation of genes in prokaryotic organisms is known as the Operon model. And what the Operon is, it's basically a unit or a segment of DNA that contains two important sections. It contains the regulatory section as well as the coding section. Now, what exactly is the regulatory section and what is our coding section? Well, to see what that is, let's take a look at the following diagram. So this is basically our operon. So this operon consists of the regulatory section and the coding section. Now, the regulatory section itself consists of two important control sides. We have the promoter side as well as the operator side, and we'll see what the function of those sides is in just a moment. And our coding section actually consists of sequence of DNA that code for certain types of polypeptides, for certain types of proteins. So this is our operon. It consists of our control sites, and it also consists of the structural genes. Now, any given unit, any given operon itself also is found next to a regulatory gene. And the regulatory gene basically consists of the sequence of DNA that codes for the repressor or activator. So every single operon next to the operon contains the regulatory gene that codes for either the repressor or the activator that is involved within that operon. Now, to gain more intuition and understanding to what the operon is and how it actually works, let's take a look at one particular example of the an operon known as the Lac Operon, which stands for the lactose operon. This is the operon that is used by a certain type of prokaryotic cell known as E. coli. So prokaryotic cells basically control the genes by using the change in concentration of certain types of biological molecules. So prokaryotic cells have genes that respond to changes in concentration of certain types of biological molecules, such as glucose. So basically, when glucose concentration is high, the glucose can basically, or the cell can metabolize the glucose and use the ATP produced by the process of glycolysis to power the different types of processes that take place within our cell. However, what happens when the glucose concentration is low. When the glucose concentration is low, the cell must actually metabolize some other type of biological molecule, and one other common type of biological molecule that the cell metabolizes is lactose. So lactose is a sugar molecule. It's a disaccharide that consists of two individual sugar monomers. So our lactose consists of a glucose as well as a galactose. Now, the enzyme that actually catalyzes the breakdown of lactose into glucose is known as beta-galactosidase. And beta-galactosidase is basically controlled. So the synthesis of the protein, the enzyme beta-galactosidase, is controlled by a special type of operon found on the DNA of the prokaryotic cell known as the Lac Operon. So let's take a look at the Lac Operon. The Lac Operon is basically this section here. So the Lac Operon consists of two sections. We have the regulatory section, which is basically this section here, and we have our coding section. The coding section basically consists of 3 genes that each code for a specific type of enzyme that is involved in the breakdown of lactose. So this region, shown by region number 6, contains the gene for the beta-galactosidase, while these other genes are genes that code for 2 other proteins, 2 other enzymes involved in the breakdown of lactose into glucose and galactose. Now, what about this section? Basically, this section is the regulatory section. It consists of our control sites. Now, the Lac Operon actually consists of 3 sites. We have our operator site, we have the promoter site, and we also have another type of site known as the cap site, and we'll discuss what that means in just a moment. And to the left of our cap site, of site number 3, we have the gene that codes for the repressor protein, and we also have the promoter of our gene that codes for the repressor protein. So let's discuss once again what happens when our cell contains a high concentration of glucose. So when glucose concentration is high, RNA polymerase will bind to the promoter region, so region number 1, and when RNA polymerase binds to our promoter region, it will basically transcribe this DNA molecule into an mRNA molecule, and then that mRNA molecule will be used by the ribosome to basically synthesize the repressor protein. And once our repressor protein is formed, it will go on and bind to site number 5. So site number 5 is one of the control sites. It's known as our operator region. And once the repressor protein binds to the operator region by using electric forces, once it binds, it basically will block the process of transcription of these genes here. So once formed, the repressor protein binds to the operator region and blocks the RNA polymerase from transcribing the mRNA molecule that codes for the beta-galactosidase, as well as these other 2 types of genes. Now, why exactly does the cell need to block the transcription of these mRNA molecules that code for the proteins that break down lactose? So this is because inside the cell we have a high concentration of glucose. So we do not actually have to break down lactose because we have the glucose that we can use as our energy source. And so because the concentration of glucose is high, we do not need to synthesize the proteins, the enzymes that are involved in the breakdown of lactose. So once again, this takes place because at high concentration of glucose, the cell does not need to use lactose as the energy source because it has the glucose in the first place. And so the genes for our beta-galactosidase, as well as these other two genes, the proteins involved in the breakdown of lactose, are essentially turned off. They are not expressed. Now, what exactly happens when the glucose concentration drops? So when the glucose concentration drops, our cyclic AMP, or cyclic adenosine monophosphate concentration, will increase, and our cAMP molecule will go on to bind to a special type of protein known as our catabolite activator protein, or CAP. And what the CAP eventually does is it will bind to site number 3. But before that actually takes place, when we have a low concentration of glucose, we're going to have a high concentration of lactose. And some of that lactose will transform into another molecule known as allolactose. And allolactose is basically the allosteric molecule that will go on and bind to our repressor protein, and once it binds to the repressor protein shown in red, it will inactivate that repressor protein, and that repressor protein that is bound to the operator will basically detach from the operator, and once it detaches, then our cap can basically bind to the cap side, and that will activate the promoter region, this region that basically promotes the transcription of these genes. And so when our repressor is inactivated and our cap binds to our cap side, only then can the RNA polymerase actually bind to the promoter and begin the transcription of these genes. So at a low concentration of our glucose, we want to be able to break down lactose to actually form the glucose that we can use to break down and form our ATP molecules. And so at low concentration of glucose, we want to express the formation of our beta-galactosidase, so that we can use this beta-galactosidase to actually break down our lactose molecule. So once again, when the glucose concentration is low and lactose concentration is high, the lactose can transform into another disaccharide known as allolactose, which binds to the repressor protein allosterically and inactivates that repressor protein, which then detaches from the operator side on our Operon, on our Lac Operon. Now, at the same exact time, because we have a low concentration of glucose, we're going to have a high concentration of cyclic adenosine monophosphate, or cAMP. And then cAMP will go on and bind to a molecule known as the catabolite activator protein, or CAP, and the CAP goes on to a special site that is shown by site number 3, that is known as the CAP site. So the cap side is part of the regulatory section, and once it binds to our cap side, that will activate our promoter, and the RNA polymerase will bind to our promoter and will begin the synthesis of the mRNA molecules that code for these proteins. And then the proteins can synthesize, and once we synthesize our proteins, those proteins can be used to metabolize our lactose to form the glucose that can then be used to form ATP via the pathway of glycolysis.